Good afternoon, everyone. And my name's Paul Diehl. I'm the COO at Cellecta. And thank you all for coming, taking some time out of your day to come and see the presentations that we put together this afternoon. Before I get started, we have to show you this. In case you haven't realized, this is a exhibitor spotlight theater. It doesn't have the credits, I guess, or something. So, Selecta, before we get started with the actual presentations, let me just give you a few minutes introduction to our company. We are a small technology-focused development enterprise south of San Francisco in Mountain View, California, about half an hour south. And the company itself is focused on developing products and services for identifying the genes that matter. And so what are the genes that matter? Genes, especially for us, for human health, are ones that are therapeutically relevant as potential targets or biomarkers that can give you information about patients' health. And so we primarily focus on have developed three main technology platforms to help take the model systems that you have for different diseases and different systems that you're studying, and then identify these genes that are important for the phenotypes of those systems. And most of the readout is next generation sequencing from us all of our technology. The three key core platforms that we focus on are the functional genetic screening platform. This is really the foundational platform for Cellecta. The group that's founded Cellecta actually launched the first pooled libraries for shRNA screening. And since then, of course, we developed out that platform, but also then when CRISPR came along and CRISPR technology and the various variants, all of those fall into the same workflow. And so we have a complete platform on that, which I'll talk about in a few moments. Then we also have, as an extension of that, the pooled oligonucleotide libraries. We have barcoding libraries, and we can incorporate barcodes into the sgRNA functional screening libraries also. And then the third core platform that we have is the expression profiling, gene profiling, and immune repertoire profiling systems. And so today we're going to talk primarily about the last platform, which is the profiling systems. Our CEO, Dr. Alex Chenchek, is going to talk about that at the end. And then before that, we have one of our customers that we've been working with that has done some interesting studies with long non-coding RNA, Dr. Eugenio Morelli, and he's going to talk about the types of screening and the functional screening that he's been doing. So then just to give you a little bit more about the company again before we get to those presentations. We have been pretty successful with our approach. We've been in business for almost 20 years. We have over 600 cited publications with groups that have either used our products or used our services, or we've done some sort of collaborations with. These are ones that, we were able to identify, and they actually mentioned us in the publication. And most of these, as you can see, focus on the three core technology platforms that I mentioned in the middle: CRISPR screening, cell barcoding, or the genetic or air profiling. Most of them tend to be cancer focused. This tends to be the area that has a lot of molecular biology studies involved. And they are for, discovery of biomarkers, identification of drug mechanism of action, drug synergies, etc. So to give you an example of the approach that we take for these various platforms, we have, this is the workflow for functional screening, and this is really the full workflow from developing the libraries all the way through to packaging them, introducing them into cells, and then being able to do the analysis and identify which targets are important then. And so we support this entire workflow from start to finish. We design, make the libraries. We actually will do the screening. We have the complete capability to do this from end to end in-house. In addition to that, all of our technology that we offer, we try to make it as accessible as possible. So we can run it, we can run part of it and as services. So if you're interested in running the screening part, for example, we can then do the sequencing for you if you send those samples back to us. We can just make the library, you can take it from there. And we have a complete line of products that are supporting each one of those steps in the service. So it's the complete workflow then that we're focusing on, all the way through to the bioinformatics. And since we've had so much experience doing this, one of our core competencies is really being able to make very good quality libraries for CRISPR screening and for shRNA for that matter. They have a very narrow SKU ratio. We have minimum standards in order to make sure that they meet before we would provide them to you for screening. Since we do sequencing for researchers that are doing screens, we sometimes will sequence libraries from other sources, and you can see that a lot of these libraries sometimes are missing guides and have skewed short sort of distributions, and so this could compromise the screen, and it's unfortunately when you get to the end of the screen and you find that out. We also, as I mentioned, do start to finish functional genomics research, so we can run the entire screen for you. Also with the TCR-BCR repertoire profiling that you'll hear about later today, we run those. We can run those in-house as custom, or we provide them as kits so that you can run them yourselves. Again, we're trying to make it as flexible as possible. And so with that then, I'd like to start move over to the main talks in the presentation. As I mentioned, we have Dr. Alex Chenchak, who's the president and CSO, who will be talking later about the TCR-BCR repertoire profiling and biomarker discovery technology. But before that, we have our featured speaker, Dr. Eugenio Morelli, from Candiolo Cancer Institute and the University of Torino, and he's also faculty at Dana-Farber Cancer Institute. And he's going to talk about looking at long non-coding RNA dependencies in cancer. And so I'll turn the podium over to him. Thank you.